So in this study, we analyzed gray whale transcriptome and compared it to other species. It's slightly different from machine learning paper that had been presented previously because we had de novo transcriptome and we actually started from it. So gray whale is a hard to get animal and it's a long-lived mammal, 77 years. So we had a chance to sequence 2 tissues of liver and kidney. And we took this opportunity, but that was just to analyze transcriptome. Doesn't give us as much insight as to compare to other animals. So we choose other publicly available animals that have been sequenced already. And we choose a few of them with some thoughts in mind. So we have 2 other whales for a positive control. We have human, of course, and mouse. Mouse as short-lived model. We have naked mole rat as long-lived model that is actually outstanding because it's small and it's comparable to mouse but lives much longer. Somewhere in the process we added a bat that also long-lived outstanding animal. We took a cow as short-lived control. First of all, after we compared the annotations, what are the overlap between annotation between species. It's a big challenge to compare different transcriptomes because they've been done different times with, sorry. You can see it now, right? You can see my screen, yeah?
Yeah.
So in order to compare them, we only choose the overlapping genes that have been expressed in all 8 species that we actually compared. Here you can see the actual numbers. And of course, we reduce the number of genes that we compare, but at least we reduce some bias. One of the surprising results for us, even though it's been reported in many papers, that many of the highly expressed genes actually annotated till today. And we try to look into it with comparing the sequences, aligning them, looking into the domains, trying to predict the functions. Most of them predicted or poorly understood or unstudied, but it seems like basic functions and they might be potentially important for longevity, but in order to fully investigate this, we need to perform wet lab experiments. So as I said previously, it's hard to just compare different transcriptomes, and you can't just compare their expression levels. So we thought about another way. First of all, we grouped all the genes by Go categories. It's pretty much standard that we normalize this with TPMs. And then we ranked all the TPM counts from rank 1 being the highest expression and almost 10,000, it's the lowest expression. Here, this is the workflow or work that is not presented in the paper. You can see the small difference that the novel transcriptome of the gray whale we assembled with Trinity software and Busco for quality control, and other transcriptomes we took it directly from the database, and we performed almost pretty much standard RNA pipeline that you can see here. I won't go into too much technical details. And then we did analysis, as I mentioned, TPMs that shows us the expression level. Then we ranked them in order to be able to compare them. And in the end, we did the correlations between ranks in order to highlight the biggest differences. And we generated the heat maps. Before we even analyze the data, of course, we spend a lot of time validating it. We wanted to see, we took all the species, all 8 species for liver and kidney, and we correlated all the ranks between them and all species. We wanted to see two things. That first of all, we can compare them. They don't correlate completely between them. On the other hand, that they make some kind of sense that whales, for example, cluster together, their expression levels are similar to each other in comparison to mice, for example. Then we created the heat map of all longevity-associated goals, and it was somewhat complicated to analyze. But when we started to look into specific processes, for example, DNA repair, I think it's the most the clear picture to see. We immediately see the clustering. We saw that long-lived animals cluster together, and they correlate much. They correlate much better in terms of DNA repair, for example, and they correlate negatively with short-lived species as cows and mice. Also, in order to perform another step of validation, we took two different transcriptome cows and two different transcriptome mice. Just as a control to see that we see the same pattern in different transcriptomes of the same species. Another clear result that we got for ubiquitination, and that is well known to be involved in longevity, of course. And here we also saw the same pattern, that long-lived species correlate much better between them, and short-lived species correlate between them, but correlate negatively with long-lived species. We saw similar pattern for autophagy and some other processes that I will mention in the end. Then we wanted to see longevity genes from GeneAge database. Surprisingly, we didn't find a lot of them. That might be for many reasons. First of all, we limited the number of genes that we analyzed only to those which overlap between species. Second, here, same as in machine learning paper, we actually looked only on longevity-associated genes that correlate, that their expression level correlate with lifespan, and not just being expressed or not expressed. Here we saw that generally we found almost the same amount of longevity-associated genes, anti-longevity and pro-longevity, but in long-lived species, pro-longevity genes expressed much more than in short-lived species, and the same for anti-longevity genes. Here you can see some actual numbers of the genes from the paper. And to summarize this, so the main findings that we found, and then it's been reported in many other papers also, that high expression of genes related to DNA maintenance and repair, autophagy, ubiquitination, apoptosis, of course, and immune responses linked to be highly expressed in long-lived species. Also, expression pro and anti-longevity genes found in model organisms coincides well with their effect on lifespan. We looked a little bit into functions of pro-longevity genes, and we saw that many of them are stress-related. We concluded that in whales in particular, it might be potentially gives them ability to resist cancer and stress in the environment. Here is the graphical abstract for this. So these 5 processes were the main findings, but there are much more, of course. And we saw that all of this has decreased expression in short-lived species on the right and increased expression in long-lived species on the left. To brag a little bit about, so we already published this paper, and even before we published, it's been featured in a popular article in New Scientist magazine. And I would like to thank all the team members who participate in this research, also all the co-authors, of course, of the paper and of the lab in general. Thanks, Robbie, for Geotomics and cool research we done. Thank you for Finding Agency that actually brought me to Romania to have productive period and intensive research. Thanks, everyone. If you have any questions.
Thank you, Mitya, for a great talk. You were actually very fast, so we have time for a couple of questions if there are questions in the audience.